this one. Okay, so first, I'd like to thank the organizers for inviting me today. So I realized, I'm talking between the poster session and the drinks. So I'll try to be as fast as possible, and I may skip a few details or slides, so to make sure you have some drinks left, so for you when you go downstairs, So I'm going to touch on some points, that have been discussed today at the symposium, so the open aspects, so Phil earlier talked about, industry and academic partnership as well. So I will give you some examples, What I wanted to do is, give you a sense of, working in a pharmaceutical company, how we use data, how we analyze data, and using a few stories, so to illustrate that point, So first, I want just to set the scene, so and if you want to fail, basically don't work in the pharmaceutical industry, I guess, So because the issue we have is we start a lot of projects, so and an example you can analysis over 10 years, and to show how many I think are going to progress for what we call clinical phase 1 to the market, so and basically I think so it's less than 20% on average, so it's it's a graveyard of projects, so and so a lot of people have been looking why, I think, because of course that's a big issue, and the many analyses, that's one I think was published now 2-3 years ago. And all of the analysis, seems to point at the lack of efficacy is the main cause of what we call attrition, so losing those programs or projects. There's other reasons as well we need to take care of, safety, strategic, commercial, so on and so forth, so but efficacy is the main one, so that's the reason why we want to ask those kind of questions, so how we can stop this, so and if it's efficacy, how maybe we can pick early on the right targets, of course we have to think of the right disease and the right patient, so but thinking so that so the avoiding, I guess, to go with the wrong target for a long time, so and I work in computational biology, we're thinking about what methods, what approaches, what data we want to use. And when it comes to data, I think in fact we use a lot of external data. We do generate data, but we use, especially in the discovery space, a lot of external data. So it's not like we are short of data necessarily, I think, and that's the example of DNA sequencing here. And I just want to point to the human, and Phil talked to that about earlier. And I think he was right, so I'm old enough to remember that when the human genome came, so in 5 years later, we'll be out of jobs, because all the drug targets will be found, so, and of course, that's not the case at all, I think, so I will point out, I think we may not be short of data, but we may not have always the right data. I think that's, maybe I think something to think about as well when people do generate data, making sure the data address the questions, so we have in front of us. And of course, the progress in use at sequencing, so are linked to technology, progress in technology, and I think that's an important connection, data and technology, progress in automation, costs, of course, with the next generation sequencing, so the costs have been reduced, and you can generate a lot more data. In that kind of graphs have been shown, many times, the growth of data. But it is more than one technology. I think if you look back, the last few years, you have single cell, for example, you have CRISPR or gene editing in general, CRISPR, talent, things like that, So you have the combination of those. You have the progress, in terms of compute powers, so methods as well, So we heard about in different sessions, about AI, ML, and I think that combination of this, so technologies, data generation, computing, make it extremely exciting, so for us. So this is just try to illustrate the way we are thinking about it, at least at GSK, so we try to combine. I think we describe a little bit we feel the importance of human genetics, so combined with functional genomics, we have really increase in terms of AI, ML as well, so our capabilities. But a lot of it as well is through collaborations, and here are some examples of collaborations we have. accessing data, so UK Biobank, for example, with a collaboration with 23andMe as well. I'm going to show you some examples today of open targets, which is a big, industry partnership, so that we initiated with Humble ABI and the Sanger, and has grown over time, so we have 5 companies now, and hopefully we have a 6th one in before the end of the year as well. So, and I think that's very important, this public industry partnership, so and the fact, it's called open targets, because we want to make the data or the methods, available as well. So what are we trying to do? It's kind of engineering, in fact. We want to really to understand, so our engine, and what I mean by that, so what's happening in our body in case of health or disease, so, and really get to a deep and causal understanding. That's all very, very nice, but it's not like building a plane, so it's or things like that, so it's a lot more complicated. So you have the input. Okay, you have your genome, genetics, so, but you have the environment, your lifestyle, so your diet, and that create an output, so, and you have multiple layers as well. So you have the metabolome, the transcriptome, the proteome. You can measure things in different ways, and all those different layers are influenced as well by the environment, for example. There is a question of scale as well. So, you have to look, you look at this, and it's multi-level, look at the cell, the tissue, the organ, the organism, so, and something that I feel is good, it's, we need to pay attention to, I think, probably more the way we generate data as well, the temporal aspect, the dynamic aspects as well. So, something that we're trying to look into in terms of data generation and method and data analysis as well. So, disease are very complex as well. So many diseases are complex, so it's interesting as well that a lot have been defined a long time ago. And I think, so what we're learning as well is like, so that one disease doesn't equal to one mechanism, for example. There is a lot of differences, so between people. And I think, if I look at genetics, and this is starting to change, so, but there is really a lack of diversity in terms of data. A lot of the data in genetics as I came, from in general from white Caucasians, so, and that's something that I feel need to be addressed as well. So if we really want to understand disease and across different populations, there's a genetics to start, so, but of course, there's a cell interactions, so which cells you really want to go after. For example, in terms of disease treatment is something important. So it's very, very, very complex, So we started, so a few years ago to think about, genetics, despite the limitation in terms of diversity, and the way it came across, it's a little story here, so there was a paper published by Francis Collins, over 10 years ago, where it was shown that, so there's quite a few, it's called GWAS loci that had, so some drug targets, in fact, so. And a year later, I think we were doing some work, in fact, on drug repositioning using GWAS genes, we call GWAS genes, and we did notice that there was some form of enrichment in terms of drug targets as well. So, and again, a couple of years later, some colleagues at AstraZeneca demonstrated that their programs to be more look to be more successful in phase 2, where the one had some genetics evidence as well. So and the other one were failing. Then we published in 2015 a paper in Nature Genetics showing that so again, if you have some genetic signal and we look at GWAS, we look at OMIM, something like that, you have maybe a twofold higher chance to have a successful drug target. So that's why I think we've been spending quite a lot of our time and resource doing some genetics, using genetics data. So more recent work, again, collaboration with Open Targets was published in November, I think, last year, looking at FDA approved drugs in 2021, and that just confirmed the story that so quite a new drugs approved, so 50 drugs, so were approved, and 2/3 had some evidence of human genetics. And there was, in fact, 2 weeks ago, there is a paper in Med Archive, again, giving the same message. So that's good. We use a lot of genetics data, but genetics get you only so far, I think. So we need really to understand the mechanisms as well. And that's why we start to look at using functional genomics. One example here, I think the type of things we do in terms of data generation, you may have some, identify a target with some genetics evidence, and that's not, I'm skipping, through all the details here. It's not, what you call a causal genes, that's not that easy to find, So there's a lot of complexity as well in terms of making sure you got the right gene, so in terms of genetics. So we may want to do some preclinical, what we call preclinical experiments. We may want to activate or inhibit, the gene in a certain system. And then we come, with some omics profiling, and we generating the signatures, and trying to compare that, for example, to disease signatures as well. So there's quite a lot of work, so in that space. As I said, diseases are very complex, and what we're trying to do is, to generate, so deep phenotype, what we call deep phenotyping data to really understand what's going on with disease, So, and there will be, multiple omics type of data. Could be, I think, at cell level, tissues. Again, we're trying to, look at, are we having some naive patients, or patients that, are under some certain treatment, for example. So trying to think about diversity. So we have, various partnerships, so where we generate the data, so we're using public domain resources as well. In the US, for example, there is this AMP, the Accelerated Medicine Partnerships that started with the NIH and various companies and the FDA. So generating that type of data. And we use that, to inform or later stage projects, for example, and that's what we call forward translation. What we're doing as well is, using the GSK clinical studies by generating omics data, and in that case, trying to inform or early research as well. So what we call reverse translation. So a lot of genomics data being generated, and basically, I think trying to use it, across the drug discovery pipeline, as you can see at the bottom of that slide. As I said, we do generate a lot of multi-omics data. I'm not going to talk, lack of time, about all these examples on that slide, so as I was necessarily planning, so I'm going to focus maybe on the right, the right-hand side for you, I think, so the integrative analysis, with multi-omics, an example in the context of a model we have with collaborators in Germany. It's a tumor lung cancer explant model where we've been generating multiple omics type of data, as you can see, proteomics, so on and so forth, so and using methods developed by colleagues at Amber in Heidelberg and in Kingston, using a method called MOFA, which is multi-omics factor analysis, so trying to understand the different technical or biological factors to explain that kind of multi-omics data. And we're using that, so to understand, patients or target or mechanism of actions or for biomarker discovery. So I'm going to talk now a little bit more about networks and the way we think about using networks, and the importance of that data. So the example here is, so where we, for example, introduce some genetic or chemical perturbation in certain systems, as I mentioned earlier. So sometimes we want to perturb a target, we can use CRISPR, for example, or we want to use a compound, to perturb a target, or we want to do both the genetic and the chemical perturbation and look, how similar or dissimilar these things are, So, and again, in a system, so that we believe, and that's why, for example, we may use some genetics, to find, which cell type may be enriched, so using some genetics data. And generate these multi-omics data, transcriptomics, proteomics, metabolomics, and really trying to understand, so the networks, the causality between the genes, you know. So, and the way we think of, you know, we do that, you know. And by the way, I will, if you want to have more details on that type of work, I will recommend, you know, we have a poster, I think it's poster 127 tonight, you know, so where you can get more details on the kind of new methods, in fact, you know, so we're developing in that space, you know, to really understand the mechanisms, you know. So, but we have, you know, access to some what we call causal networks. We can look at the abundance, you know, so of these nodes of genes, you know, so using these different assays, you know, so we can quantify that. We can also, I think, you know, so move from activity, I think, you know, so up and down, you know, so, and do some hypothesis ranking as well, you know, to look at the path, for example, to go from gene A to gene J in that case, you know. So, and that's help us, I think, to again to understand the mechanism of actions, you know, maybe to find, you know, new drug targets or biomarkers and things like that. That's one way we think about networks. Again, I don't go into the details, don't have the time, you know. So, but there's a poster, you know, elaborating on that kind of things, you know. So. The other example I want to give you is a paper we published, you know, 3 years ago now. So, and we call network and pathway expansion of genetic disease association. And the way we are thinking about it is the fact that we can have, you know, what we call a GWAS genes, you know, gene we are confident is the gene given the genetic with a genetic signal for a certain disease. But for example, that gene may not be very tractable, you know, by a small molecule or a biological, for example. So we've been looking at what we call proxy genes, you know, genes that have some form of relationship between that you know, with that GWAS genes, you know, and can that genes be a new target, for example. And the way we looked into that, you know, was doing enrichment again, you know, looking at you know, and using benchmark, you know, in terms of successful drug targets. Using a database called Pharma Project in that case, you know, so, and been looking at various type of resources, you know, so like protein complexes, you know, ligand receptor database or network pathway databases, and maybe not surprisingly, so we found, you know, there's error bars could be quite large in some cases, but we find some enrichment, you know, when it's a protein complex, maybe like the same function, and also with ligand receptor, you know. So again, you know, maybe not too surprising, but that give us a sense, you know, maybe looking at what we call these proxy genes can give us, you know, so potential, you know, new targets, for example. And I'm going to, and sorry, I'm rushing to look at the time. I think the poster session has started, but I'm going to finish, you know, so by a very recent example of work that was published, you know, with our colleagues at Open Targets in, again, I think it was Nature Genetics, you know, so earlier this year, and trying to really to look at, you know, so a network in the context of human genetics, you know, so. And again, that's important for us, you know, so because that can, you know, help us to understand the biological processes. You can look, as I said, you know, for new drug targets, you know, so, and could help us as well to predict, you know, some maybe causal gene, you know, with that locus to gene function. So the way it was done, you know, so what we needed to have is, you know, and collaborators needed to have is an interactome. And in that case, you know, so we use a string, but also another open target project, you know, so where there was a combination of intact reactome and signal, and we combined this OTAR project with string, you know, so, and you can see the, you know, it was over 500,000, you know, edge counts, you know, so, and the number of nodes, genes, the focus, you know, so on that OTAR project combining intact reactome and signal was human data, so we didn't use non-human data. So that was our interactome source, you know, that combination. And we needed as well to have, you know, so the genes, you know, so coming from GWAS studies. In that case, we use, again, another resource that was developed by Open Targets called Open Targets Genetics, so which basically help you to map, you know, the SNP to what is the most likely causal gene for that disease, you know. So we had that list of genes coming from the Open Target Genetics, and by the way, that's all, you know, available. You can look and search, you know, so that platform. Then, you know, using that those two things, you know, so we had the interactome mapped, you know, so the genes, you know, so on this interactome did some network propagation, clustering, and I think the modules as well, and significant modules with this approach, you know. So I'm going to skip the bottom here, I think, so, and go straight to the way we kind of been using that data. If you look at, so some of these networks, what you can start to look at, so you can have different traits coming from the GWAS, and you can ask, the questions, I guess, so using the network propagation, for example, I think, so what are the similarities you see between different traits, so, and using pairwise, similarity approach, so you can do some hierarchical clustering and start to build, I guess, so what are the relationships between those diseases, and can tell us, maybe what are the functional, similarities between diseases. And here are just two examples, so of skin diseases at the top and immune disease at the bottom. And that could be quite important for us, so for example, in the context of if we develop a drug for a certain disease, so maybe I think, we can look at doing some repositioning for a different disease, for example. So that's one way we can use that kind of data, or really again, understanding, I guess, so the similarities. The other example, the way we want to use that data is, to look at common mechanisms, so more in the case of pleiotropy, for example, so where we have, different traits, so, and look at these modules we build, and see, if we have more than one trait per module. In fact, we found, 73, I think it was 73 modules, that had more than at least two traits, so. And again, that's quite important for us to understand, because that can lead to potentially common mechanisms. And here is just one example I want to give you, bone and fasciitis, so, and you can see the clustering, and that's a module on the right hand side. And what you can start to do is also annotate that module, so for example, so there was a drug, romosozumab, that was targeting one node, one gene in that module. We also, in blue, you have, for example, I think we annotated with a mouse phenotype, mouse knockout phenotype for some of those nodes in the network, and you can see abnormal kidney or abnormal heart morphology, and that's tell us, and maybe I think so there's a risk by developing a drug here. I think there may be a safety signal for that. So we did annotate as well with a ClinVar the genes, and some were not identified by GWAS. So we can really start to play with those networks, and understand the mechanism, the risk potentially, the disease expansion, so how we approach it. And we keep building on that work, with our colleagues at Open Targets, especially with colleagues at ECCB, for example, so we're trying to build, networks now would be more, for example, cell type specific, rather than more general as well. So that's another Open Targets project that is ongoing in that space. So I'm going to conclude now by just telling you that I feel it's a very exciting and unique time, despite the challenges. As I said, we have data, maybe not always the right data. I said we're not really short of data. We have all those methods, so that are developed as well, the technologies that are coming with it as well. So I guess the only thing I will say, I think, and maybe because I've been in that business for many years, is all very nice, but I find that sometimes more data, more technology, then basically biology becomes more complex. And that's what I've got this onion here. I think so feel it's like peeling an onion. I think there's layers and layers of complexity, and I'm not sure it's much easier, I think, to find a new drug, that it was, necessarily 10 or 15 years ago. But that, I think it's exciting, so because we probably understand better as well, so what we can do and what we cannot do. So, but it is, biology remains challenging. And I'd just like to conclude, to acknowledge, the different team at GSK, especially computational biology team, so that some of the work has been presented here, but our collaborators, and especially, I think, the work today are presented, Open Targets and EMBL. Thank you.